In the last part of this math and stats review, we're going to take a look at the chi-square test. Chi-square, if you remember, is a statistical test that we use to see if what we got was close enough to what we expected. And if you think back to when we used this when we learned about mitosis and we compared the number of cells that we counted versus the number of cells that we expected to see if what we got was close enough. We also use this with our genetics problems, so looking at Punnett squares, and again, looking at real data of a genetic cross. Did the number of offspring that we got match the expected ratios? So that's what we're looking at with a chi-square test. I'm going to walk you through a simple example, and these notes you have seen before, but I'm just going to reuse them to help you remember what this chi-square test is used for. So we're going to look at flipping a coin 100 times and seeing if our coin is fair, right? So in order to be fair, we would expect that we would get 50 heads and 50 tails if we flip that coin 100 times. Now remember, with a chi-square test, we always start with a null hypothesis. The null hypothesis is always one that's this option. Okay, so there will be no difference between the observed data and the expected data. We kind of make the assumption that what we get is supposed to be close enough to what we expected. Okay, so that's our null hypothesis. Null, think no difference. So here is a good one, another example for the null hypothesis. If we wonder if world-class musicians have quicker reaction times than world-class athletes, remember our null, no difference. There will be no difference between these two groups of individuals. Okay, so kind of always use that same sentence starter. There will be no difference. All null hypotheses start off like that. Now let's go back to our flipping coins. So let's say that you flip your coin 100 times and this is your observed. You get 41 heads and 59 tails. That's kind of close. It's kind of close to 50-50, but how do you know if it is close enough, right? Some people might say, yes, that's close enough, and other people might say, no, that's not. It's just kind of far off. That's where the chi-square test comes in. It gives us a statistical analysis that says either yes, our data is close enough to what we expected, or no, definitely not, okay? So you might remember this formula. This formula is on your formula sheet where you take the observed minus the expected, you square each of those answers, divide each of those by the expected, and then add all of that up. Now again, you might look at this equation and not remember using the equation, but you might remember using a chart that looks like this, right? So we have a column with observed, the number of heads and tails, the number of expected, what we thought would happen. And then we take our observed minus expected for each row, square that, divide each by the expected, and then remember this symbol means sum. So we add those numbers together. So if you would see a chi-square test on your AP exam, I would suggest making a little chart like this. And again, remember, you can have any materials in front of you while you're taking the test. So it might be helpful to have an example problem, maybe this one, so that you remember exactly how to set up these types of problems. Okay, so now we have our chi-square value. We have 3.24. So what? What does that tell us? Does that tell us yes, we're close enough, or no, we're not close enough? That's where this chart comes in. And again, if you'd like to have this chart in front of you while you're taking the AP exam, that is more than allowed. That is allowed. Okay, so two things that we want to look at. We want to look at the degrees of freedom, and we want to look at the probability value. Now remember, as biologists, we only use the 0.05 column. That gives us a 95% confidence that we like to use. So we always use that column. So first, let's use our degrees of freedom. The degrees of freedom is determined by taking the number of options or the number of groups that we had minus 1. So our two options for our coin flipping experiment, we had either heads or tails, right? So there's two options. So we would take 2 minus 1, and our degree of freedom is 1. What does that mean? It means that we're going to take that number over to this column, and that's the number that we're going to use in our comparison. Okay, so again, we always use this column, and the degrees of freedom just tells us which number to use. Okay, so again, now we have two numbers. Now what? Our p-value is 3.84. Our chi-square value was 3.25. What the heck's that mean? So we compare those two numbers, our value that we calculated from our chi-square chart, and then the number that comes off of the probability chart, and we see which number is bigger. If our number that we calculated is smaller than the p-value, we accept our null. That tells us that the number of coin flips that we got was close enough to what we expected. There is no significant difference between what we got and what we expected, right? So the differences in between our data is just due to random chance. Now, what if, on the other hand, we got really crazy numbers, like we got 30 heads and 70 tails? Those numbers seem a little bit off, right? So we would expect that our chi-square value would be higher, and our number that we calculate would be higher than our p-value. That tells us then that our coin flip experiment, our data that we got, our observed, was not close enough to our expected. So there was something going on with that coin. Okay, so that's just a quick refresher of how to do a chi-square problem. Now, this is a practice problem, taking this and applying it to genetics. So pause the video here, take a minute and read this problem. And this first part is having you do a Punnett square. This is going to be a dihybrid, so one of those bigger ones, Punnett square. So take a minute, pause the video, figure out your Punnett square, and then we can take a look at the answer here in a second. So let's start our Punnett square by looking at what the parents are. The first parent is dihybrid heterozygous. So that tells us that this plant is heterozygous for both traits. The other parent is homozygous purple for stem color and heterozygous for the leaf trait. So let's kind of work that out over here. So if parent number 1 is double heterozygous, that plant is going to be big P, big G, little g. And parent number 2 was homozygous purple, so big P, and then heterozygous big G, little g. Now to set up your big dihybrid Punnett square, remember we need to figure out the parents' possibilities of their gametes. So for parent number 1, through meiosis, this big P and this big G could end up in a gamete, so that's one combination. Our little p and little g could end up together. We could see little p, or big G. And then which one am I missing? The outer. We could end up with big P, big G. So remember we learned that FOIL method. That's how we figure out this parent's possible gametes. Now let's repeat that for parent number 2. So big P, big G is one combination. Big P, little g. Big P, big G. And big P, little g. Okay, so if we fill out then our big dihybrid Punnett square, that would be the answer to our first section. So part A of that problem, you're going to cross those parents. Now remember, when you're filling out this Punnett square, you want to keep the P's together and you want to keep the G's together so that it's easy to determine what is this offspring going to look like. So we can see that it's going to be purple and green, right? Now to save some time, I'm not going to go through every single offspring because on the next slide back here, they give you the answers, right? So here's what your Punnett square should look like. And from this, I've created and looked at what we should have gotten. So this is what we would expect. So we should see 12 plants with a purple stem and green leaves, 4 plants with a purple stem and yellow leaves. So if we write that as a ratio, that's a 12:4 ratio or a 3:1 ratio, right? If we reduce that. Now that is going to help us figure out our second part of the problem. The second part of the problem is looking at, now we have real data. Now we have, okay, we actually crossed these plants, and this is what we got. This is our observed data. So we got 234 plants that were purple-green and 42 that were purple-yellow. Does this match what we got? And again, anytime they ask you something like this, and notice they're not using the word chi-square, but they're asking you to figure out, is what we got close enough to what we expected? You're going to do a chi-square. Okay, so again, I would pause the video here, use this data, create a little chart, and use our expected ratios to figure out if our data was close enough. So fast forwarding to the answers, here is my chi-square chart. Now, one thing that I've seen students struggle with this year is getting the expected. Okay, so we have a ratio. How do we turn that into data? How do we fill out our expected column? Well, remember, just copy your observed down, and we want to find the total of that. And then using this total, we're going to figure out our expected. So if we expect 3 out of 4 to be purple green, we would take 3/4 times this, or 75%, 0.75 times this. That's where this number came from. And we expect 1 out of 4 to be purple yellow. So 1/4 or 25% or 0.25 times this number is going to give us that expected. So that's how we would figure out our expected based on this data. And we end up with a chi-square value of 14.09. Now using our chart, we're going to have 1 degree of freedom using 3.84 as our value. So looking at this number, our value is way higher than what our p-value was. So the data did not support our hypothesis. So there is some sort of statistical difference between the plants that we got and the plants that we expected. Okay. In this video, there's one left, one more practice problem. So I would pause here, try this, and just to check yourself, the answer is on the upcoming slides. And then that wraps up our stats review. If you have questions or you'd like to go over anything more specifically, email me or attend our Zoom on Thursday, and you can get some more feedback with that. Thanks, guys.